Now let's take a look at heme degradation. The best way to start with heme degradation is to start at the beginning with red blood cells. The erythrocytes, of course, have a longevity of approximately 120 days. At that point, they become glycated, and then they are recognized by splenic macrophages. These splenic macrophages bind and internalize the senescent red blood cells, and their job is to break this down, digest the cells, digest the contents, and essentially recover the iron for recycling for new erythropoiesis. So once we have the red blood cell inside the macrophages, we have protease activity that degrades proteins. It releases the heme. So if we release our heme structure, let's draw our heme as a square. We know that it has 4 pearl rings to form the porphyrin ring structure. And inside we have our iron. So the first step of heme degradation is that the iron must be removed. And so the iron is extracted from the heme ring structure inside the cytosol of the macrophages. And this heme binds to hemosiderin. Hemosiderin is an iron storage protein. And this iron storage protein is analogous to what we'll see in liver called ferritin, but hemosiderin is the iron storage protein for iron in macrophages. The remainder of the heme ring structure then undergoes a series of reactions, redox reactions. The first enzyme is heme oxygenase. And heme oxygenase is a redox reaction that also introduces oxygen. It's also the only enzyme in the body that produces carbon monoxide as a byproduct. Obviously, this is very small amounts, so it's not harmful in any way. So the heme structure, once we do this, we are going to break the ring. And the redox reaction occurs at the top of the ring, and what we essentially have is the elongated porphyrin ring structure, now no longer a ring. It has been broken up. We've introduced oxygen, and we have our 4 pearl rings as part of this structure. This is called biliverdin. The next processing event is done by the enzyme biliverdin reductase. And this, of course, is another oxidoreductase enzyme. And so we are going to further reduce the biliverdin structure into a modified structure. We still maintain our pearl rings, but essentially what we've done is we've taken this center carbon and we've reduced it. This structure is called bilirubin. Now at this point, the bilirubin is secreted from the splenic macrophages and put into the blood. From the blood, the bilirubin travels to the liver, and it's taken up by the hepatocytes. The uptake of bilirubin by the hepatocytes is done by a bilirubin transporter that is ATP dependent. So we are coupling ATP hydrolysis with bilirubin uptake to ensure that we have sufficient energy to make sure we take up all the bilirubin we can. The essential nature of that is because bilirubin is highly hydrophobic and it's very toxic to cells because of this. It'll bind indiscriminately to proteins because of its hydrophobicity and inhibit activity. What prevents it from harming anything in the blood, of course, is the major protein in the blood that carries hydrophobic moieties, and this is albumin. Now, once the bilirubin is taken up by the hepatocytes, we are going to begin to process this to form one of the bile components. So the first step we have here by the hepatocytes is we are going to take 2 UDP glucuronic acids structures, and we are going to conjugate this to bilirubin. To form bilirubin diglucuronide. So again, the structure is very similar to bilirubin. We have our parole rings here. We haven't lost those. But in this case, we've added our glucuronate residues on those positions. And so now we have gone from unconjugated, indirect bilirubin to conjugated. Conjugated is considered in lab tests as direct bilirubin. Now this bilirubin, of course, we know from our liver and gallbladder study, is secreted as a bile component. Upon CCK stimulation of the gallbladder, relaxation of the sphincter of Oddi, the bile components are now placed into the small intestine. More precisely, into the duodenum. The purpose, of course, for the bilirubin to be placed into the small intestine is to dispose of this in the stool. Now, we have in our small intestine, especially at the distal small intestine, we have some bacteria. Now, what the bacteria are going to do is to deconjugate the bilirubin diglucuronide by taking off these glucuronic acid moieties. And so we will be returning the structure in the small intestine as bilirubin. And this is called the deconjugation. Once the bilirubin is in the small intestine, after it's been deconjugated to the indirect form, it is now going to be taken up, or modified by bacteria further. And the modification of bilirubin are simply, the majority of these reactions are redox reactions, so there's very simple modifications. So the bacteria will take the bilirubin, convert it into urobilirubin, also called urobilinogenin, and then further reduction to go from bilirubin to urobilinogen to stercobilin. Now once we have stercobilin, this will end up in stool. The urobilinogen can be taken up by the terminal ileum. Small amount of it is taken up and returned by the enterohepatic circulation. So it's taken up by the terminal ileum, returned into the circulation. Now there's two places, two fates for urobilinogen. The first is that it can be taken up by hepatocytes, put back into the bile, and taken a second pass through the gallbladder and the biliary secretions. The other option it has is to go to the kidney. The kidney will further modify this by a redox reaction to make urobilin, and then this will be secreted into the urine. Now, the final point to keep in mind here is that these pearl rings, as well as the carbon linkers between these, have unsaturated bonds. That means there's lots of double bond structures in the bilirubin coming from the heme molecule. When you have double bonds, if they are conjugated, that means that they alternate, they can produce color. So hence why, if we receive an injury where we have a subcutaneous bleed that turns into a bruise, that bruise turns from a green, initially a green, which is biliverdin, to a blue-black, more bilirubin, through the oxidative state. When the urobilinogen is converted into stercobilin, stercobilin is brown. So the stercobilin is giving the stool its predominant brown color. And as you can guess, the urobilin is yellow. And that's what gives the urine its yellow color. So the color of bile, the color of stool, and the color of urine are largely due to the degradation products of heme.